All right. So again, today, for those of you that just logged on, you're going to need a few things. You should have a copy of this document. If you don't have a copy of it, I have placed a copy of it here in Google Classroom. It's this first one right here. So you can try to print it out real quick. You are definitely going to need scissors, glue, and a couple colored pencils. Okay. Also, you're going to want to go ahead and start making your way to Google Classroom. Because to start today, I'm going to have you guys on your own watch short, like it's 2.5 minutes or something like that, section of a video that I posted in here. So today we're going to be talking about lecture 6.6, and we're going to be focusing on viruses and bacteria and what we've learned and some of the things they do and some of the molecules they use that we figured out that we can use as tools. So from here on out in this unit, we are focusing on things that we need, things that we can use to manipulate bacteria and viruses for our own good. Okay, so we learn about things so that we can maybe use them or use their parts and use their molecules to benefit us. We also study them so we can figure out how to kill them. Okay, so that was kind of what lecture 6.6 was all about. So to start, what I want you guys to do is on Google Classroom is click on Tuesday, and when you get there, I do have a start and a stop time for this video because it's 10 minutes long. I don't want you to watch all 10 minutes. But the thing about this video is it's incredibly relevant because our lesson is incredibly relevant today because we're going to be talking about viruses and how they pick up mutations and how bacteria pick up mutations and stuff like that. Okay. So you're going to start at basically the 3-minute mark, and then you're going to go to 5:25. And it's this video right here that's where do new viruses come from? Okay. So I want you to go and click on that video, fast forward to 3 minutes. And I'm going to give you 2.5 minutes to just watch that small section in that video. Go for it. All right, looks like we're ready to go on. Okay, so I like this video. It was it was very relevant, and basically we're just focusing on a couple things with viruses. Is you just need to be able to tell me that whenever they're copying their DNA, replicating it to make more viruses, then they make mistakes. And so when they get those mutations, that can give them new traits. I can change those shapes of those proteins, which I'm trying to figure out what the video called those receptor proteins on the surface. But it can change the shape of them, and that gives them the ability to infect cells that they weren't able to infect before. Okay. All right. So random mutations can give viruses new trait, but then I just want to briefly mention that they mentioned reassortment recombination can cause viruses to very quickly gain new traits. And that's when you have two viruses that can infect the same cell. So they put both of their genetic material inside of the cell, and they create viruses then that are a combination of the genomes of those two viruses. This is how you get very quickly a super virus. And just note, there's only certain viruses that can do this. COVID can't, thankfully. That's a good thing. But unfortunately, one virus that's really good at doing that is the type that is our flu virus. And so if we have a situation where someone has the human flu and they're around birds, they get infected with the bird flu at the same time, then that person just created a super virus that can be transmitted easily from human to human, but then also can have very deadly effects. It's pretty crazy, but be glad it was COVID that was going on with this latest pandemic. Because what's crazy is there's other viruses out there like that H5N1 that I have on my screen right now. Think about this. The mortality rate of anyone that catches that type of flu is 60%. Like, wrap your mind around that. If you catch it, the mortality rate is 60%. So thank goodness, we've had some spillover from birds to humans, but that virus isn't able to then acquire enough good mutations in humans to be spread from human to human. And so it kind of stops in the first few people, or the first person that has contracted that avian flu virus. So I'll tell you what, though, if that if that bird flu spills over into a pig, and then from there it mutates into a human, then you probably have a virus that can be spread from human to human. This is the one. This is the one I'm really worried about. So we know that this happens, and we have to study these organisms again, so we can figure out how to stop it whenever things like this do happen, and they will continue to happen. Okay, so just one more thing kind of about viruses here that I mentioned in the video. And we're going to talk about the lytic and the lysogenic life cycle of a virus. And notice here on my screen, I have a bacteriophage. You guys have seen those before. Those creepy little spiders. Now, viruses can have two different life cycles. And actually, I'm just talking about bacteriophages. So what we see here is this bacteriophage has put in a piece of its nucleic acid inside the cell. And then what commonly happens is there's also enzymes there that will degrade the host cell's DNA. So basically kind of destroy it there. And then this cell gets busy using the ribosomes and the molecules of the bacteria cell to produce more viruses, okay? So it doesn't take long for this cell to produce a bunch of viruses. And then those viruses are going to exit the cell. And when they do so, they destroy the cell. So what you're looking at, that was a lytic life cycle. Now, to help you remember the difference between lytic and lysogenic, is think of lytic as a short word. So once this bacteria cell got infected with that virus, it's a short period of time for that cell is dead, okay? Now, the lysogenic life cycle takes a lot longer. And this is a situation where the virus has put its genetic material inside the cell, but the cell doesn't start immediately, or the virus doesn't start immediately causing the cell to make virus particles. That's what happens, is that viral DNA gets inserted into the host cell. Now, what the host cell is going to do is going to keep being a bacteria cell. And bacteria cells ultimately divide. And now we have two bacteria cells that have that viral genome in it. Now, this is kind of, so this is going to take a while. So we're not going to get any viruses produced, but that bacteria cell is just going to divide. And then you have a whole bunch of bacteria cells that are all infected with that virus. So it's reproduction of the virus, but it's over a longer period of time. Now, what can eventually happen, though, is what this virus is basically doing, even though it's not consciously doing, is this virus is waiting for a change in the environment, so that the conditions are really good for the virus to be able to spread to other cells. And whenever the environment changes, then that activates viral reproduction in all of these cells. And then the cell goes into the lytic cycle. Okay, so the viruses can go between a lysogenic cycle, and then a trigger can cause it to go in the lytic cycle. And after the lytic cycle has been triggered, it doesn't take long for that bacteria cell to die. Okay. So that is pretty much what we talked about with viruses. And now we're going to kind of switch gears here, and we're going to talk about bacteria and how bacteria can get new traits. Now, we know that there can be mutations in their DNA. When they copy their DNA, they can copy it incorrectly. But now we're going to focus on three other processes that you need to know very well. So we have transformation, conjugation, and transduction. And since we just talked about viruses, we're going to start with transduction. Now, in your notes, I found a picture of a ducking duck to maybe help you realize what's going on when we say transduction. Okay, so duck, a virus is coming. So in transduction, bacteria cells have the ability to get new traits, and it's because of a viral infection. So here's where we have a bacteriophage. I'm going to fast forward a little bit because this is going a little slow. And that bacteriophage puts its genetic material. The cell destroys the bacteria DNA as well. And then the cell, obviously the cell's in a lytic cycle, and it's making new viruses. Now take a look what happened right there. So when these viruses were being made, some bacteria DNA got put in one of those bacteriophages. So that's actually genes from a bacteria. And whenever this phage then infects the next cell, it didn't put virus genetic material in there. It put bacteria genes in there. So this bacteria now has brand new bacterial genes that can give it new traits. So this is called transduction. Whenever bacteria get new traits because of a virus delivering some new genetic material. Okay. All right. Now, the next one is transformation. And you guys already know about transformation, even though you maybe didn't know that you know about transformation. You studied it whenever we took a look at Griffith's experiment. So remember, we had the smooth and we had the rough bacteria, and one caused those rodents to die and the other one didn't. And Griffith figured out that there was something from one bacteria that was going into another one, giving it a new trait. And so that's what his experiment was all about. It was actually all about transformation, even though we didn't have a name for it yet. So this is basically what happened, is he had those two types of bacteria. And remember, in one part, he killed the deadly bacteria. And whenever you kill a cell, it breaks down into parts. And so here we now have pieces of the DNA that's now just floating in the environment. And if you mix that in with our harmless bacteria, then we knew something went into those harmless bacteria, and it was DNA. So this right here, this is transformation. Bacteria have the ability to take in DNA that's in the environment. And so now this bacteria cell has new traits. It can make new proteins. In this case, it can make proteins that were deadly to the mice. Okay, so that's what transformation is. We have transduction, duck of viruses coming, transformation. And by the way, this is the PGLOW lab that we're going to be doing this week, hopefully, if we ever get back to school. You guys are going to mix DNA with bacteria, and you're going to add some other things to help that bacteria help the DNA get into the bacteria cells. And then we're going to transform them. We're going to give bacteria new traits in our class, in this lab we're going to do. Okay. And then finally, the last process that can give bacteria new traits is conjugation. Now, honestly, I've never had a foreign language because my high school is so small that we didn't offer a foreign language. And so that was waived for me at Mizzou. I didn't have to take 2 years because the year that I was going to school was the year that they put in that requirement. You had to have 2 years of a foreign language to get into Mizzou. But I have heard enough about speaking Spanish that you have to, is it conjugate your verbs? Is that right? What does that mean if you conjugate? Okay, so to put it in, right, is kind of where you started with that. And so in conjugation, that's what's taking place here. Is bacteria are able to take pieces of their DNA, copy it, and put it into another bacteria cell. So it's called conjugation, right? So this is about this is slightly R-rated here because we're looking at, technically, we're kind of looking at bacteria sex. So this little structure that forms there is called a sex pili. And what happens is they connect via that sex pili. And this extra piece of DNA here, which is called a plasmid, which is just an extra piece of DNA that's separate from the one chromosome that bacteria have. They're very short, they're not very big. And so this one is kind of showing it. This plasmid's really big, but it's not really, they're not really that big. And this cell is copying its plasmid and it's giving it to this other bacteria cell. And so now it's going to have some new genes. It's going to be able to make some new proteins that it wasn't able to do, before it got a copy of that plasmid. So this is called conjugation. And again, it's another way that bacteria can gain new traits. Now, I do want to point out an important term so you understand what I mean. Okay. So think about what the horizon is. Okay. The horizon goes across. So this is called horizontal gene transfer. Now, someday, I would say the majority of you are going to do vertical gene transfer, which means you're going to have children. And you are here as the parents, and think of the family tree. You're going to pass your genes down vertically to the next generation. Okay. Horizontal gene transfer is where there's gene transfer between two members of the same generation. Okay. So these are examples of, or conjugation example of horizontal gene transfer, where imagine if we could do horizontal gene transfer, then I would be able to take some of my DNA and just pass it into another human, and that would give them a new trait. All right. So we can't do horizontal gene transfer, but bacteria can through conjugation. Okay. All right. Well, I think we're about ready to then move into kind of the next part of this lesson. And this is where we're going to use our models and this worksheet right here. Okay. All right. So what we're going to talk about with all that I just kind of reviewed there is now we should be able to look at, this diagram right here. And here we have a plasmid, and this is a bacteria cell. And I just showed you that bacteria cells have the ability to take in DNA from their environment. And can someone tell me what that's called? When bacteria take in extra pieces of DNA from the environment, is that transformation, transduction, or conjugation? Mm-hmm. So this is transformation, is what we saw here. But let's take a look at these plasmids. Notice they have two different colors there, because now we're going to get into how we can engineer bacteria, because we know bacteria can take in DNA and get new traits. Well, can we manipulate them and use them? And yes, we can. So what we have the ability to do is we can use tools and molecules to manipulate DNA and create it to have genes that we want it to have. And then all we have to do is take those plasmids, mix them in with our bacteria cells. They can take them in, and bam, now we have a bacteria cell that can make a protein that we want them to make. And so again, I've mentioned before that we used to get insulin from pigs and cows. So when we sent them to be slaughtered for meat, the pancreases were harvested, and that tissue was sent to a lab, and that lab was able to pull the insulin out of that tissue, and we purified it, bottled it, and shipped it to people that were diabetic. Now we don't do that anymore. We just genetically engineer bacteria to do that for us. Okay. So that's what we're going to do. We're going to show you how we can engineer bacteria or engineer plasmids that we can then put into bacteria. Okay. So let's grab this worksheet right here and a couple colored pencils. Now, what I want you to do is, with a couple colored pencils here, and this is not the time you type A people to like color in every single section of this. We're gonna just do it lightly. So grab a color. I don't care what color it is. And we are just gonna kind of shade in here these plasmids. Let me get over this. Go away. Okay, there we go. Oh, I know what the problem is. So grab a colored pencil. And what I want you to do is I just kind of want you to do a quick shade over, very lightly shade over this section, this little piece of DNA that I gave you here. And then I want you to grab a different color and do the same to this down here. And we don't have to shade in this top piece of DNA. It's already kind of shaded in anyway. And I just want you to be able to see 3 sections of this plasmid that we are going to engineer. Okay, once you get done shading, then it's time to cut out. So grab your scissors. And again, this is not the time for you to be type A. Just grab some scissors, and you're going to cut out all 3 pieces of DNA on that paper. So we'll give you a little bit of time to do that. Cut out this square, or this rectangle, and this rectangle of DNA. It's arts and crafts time today. Okay, so now we have them all cut out. And like I said, the whole purpose of this is to manipulate bacteria DNA, to insert new genes into bacteria DNA. But if you think about it, no bacteria DNA is linear. So we kind of have a problem. We need to do another step before we can even have our model set up and ready to go. So I want you to find this one right here. I colored it green. So it just says original bacteria DNA plasmid. We need to turn it into a plasmid. Remember, bacteria DNA is circular. So here's one place where we're going to need that tape. So notice that all I did is I took my bacteria DNA and I just looped it, and we're going to tape it so we can start off with circular bacteria DNA. All right. And when you get your plasmid made, throw your hands up again. And again, I just taped in the circle the one that says the bacteria DNA plasmid on it. You can see it right there. Bacteria DNA plasmid. Awesome. So now that, again, we've been talking about bacteria and viruses, then let's talk about this. And hopefully now that you're looking at this, you're like, okay, this is a bacteria cell. This is a bacteriophage. Here's a virus here. And there, that blue piece right there, that's the virus genetic material. And we can see here we have scissors, and it looks like Pac-Man because we're going to talk about this tool is something that we discovered in bacteria cells. A defense mechanism that evolved over time in bacteria cells is an enzyme that can cut virus genetic material. So therefore, then the bacteria doesn't then produce the viruses and die. Again, that's not an intentional thing bacteria did. They don't have brains. They don't have the ability to plot and plan because they're in a big war with viruses. It's just they make proteins, and these proteins happen to recognize certain sequences on viral DNA or RNA and cut it. Okay, so it's a defense mechanism that evolved over time. So we discovered these enzymes. So our scissors is kind of like those enzymes, and we have a name for them. Those are called restriction enzymes. So there's different restriction enzymes. We have found them in several different bacteria cells. And what's great about all these restriction enzymes is because they can recognize, each one can recognize a certain sequence and cut the DNA. And so they all can recognize different unique sequences. So we can use them to cut DNA where we want to cut DNA. We have to pick our restriction enzyme carefully. So what we've got here is a bacteria plasmid, and now we have scissors. This is our restriction enzyme. And you're going to need to take kind of a careful look here at my screen. Let me zoom in a little bit here if it's going to let me do that. Nope, that's a comment. Let me hide that. Hide that. There it is. Okay. All right. So this restriction enzyme recognizes this sequence right here. So this sequence is called the restriction site, and that's where restriction enzyme cuts. It recognizes GGATCC. Now, if you look up here, this restriction enzyme cuts a very unique way at this restriction site. It finds a GGATCC and it cuts between the two Gs. So this is what I need you to do. On your bacteria plasmid, I want you to look and find GGATCC. I found one right here, okay? And what I want you to do is when you find a GGATCC, I want you to draw a line between the two Gs. GGATCC, and do that on your plasmid, because then you guys are going to follow this line and cut. GGATCC. And if you don't do this right, you're not going to be able to make your recombinant DNA. Okay, so I did it right there, GGATCC, and I drew a line between. Now, notice that high and low, I think these are called like palindromes or something like that. I have another GGATCC. It's on the bottom here, right below this one. So what I want you to do on that one is, again, draw a line right here between a GGATCC. I had another one right here. And then connect those two lines. Because if we want to insert DNA in this plasmid, we have to cut it open. And we can use a restriction enzyme to do that. We discovered them in bacteria. So once you have your two lines drawn on your plasmid between the two Gs, you connect them. So mine looks like this. Cut it. You got to cut it open with your restriction enzymes. So now we've just cleaved the bacteria plasmid, and now we have a place where we can put new DNA in there. This is what do you mean by that? You don't cut it out. You find GGATCC, and then when you find that, I want you to draw a line between the two Gs in GGATCC. Okay? And then down below, there's another GGATCC. The restriction enzyme cleaves it between the two Gs. You got it? Cut it between the two. So draw a line. So I literally want you to find a GGATCC, put a line between the two Gs. Up just on my screen. Do you see my screen? Look at my screen. Okay. And then find the other GGATCC and draw a line there. Connect those two. And then cut on that blue line that I drew. Okay. So let's engineer. We want to engineer some bacteria to have some human DNA. The great thing is that because it's a universal code, which is evidence of a common ancestor, we can take DNA from one organism and we can put it in the other organism, and there's no difference between our DNA and what their DNA looks like. It's made of the same four bases, okay? So at the top, that bigger rectangle that you guys cut out, I have a sequence for human insulin, okay? Now, here's the key. We have to use the same restriction enzyme, because if we don't, you'll see that the ends won't meet up when we go to tape our human DNA into our bacteria plasmid that we just opened, okay? So if we use the same restriction enzyme, then it's going to cut at the same restriction site. So up here on the bigger piece, or wherever you have it, you guys are going to find a GGATCC. And again, between the two Gs of that sequence, I want you to draw a line. And if you find GGATCC high, you also will find one low. So find GGATCC, that's the restriction site. When the restriction enzyme finds it, it cleaves it like this. Exact same way. And you're going to find two restriction sites because we have to cut out the gene. We have linear DNA, so we can't just cut our DNA once. We have to cut on this side of the gene and make sure there's a restriction site on the other side so we can cut the other side. So we have a piece of DNA. So there's also a GGATCC on this side of the gene. So I'm going to find two Gs, GGATCC, cut down. GGATCC, I cut between the two Gs. And then connect. All right. So now we go to the human DNA and we cut out the gene. When you got 2 pieces of DNA, when you cut out that human insulin gene, just go ahead and put your hand up for me, please. All right, because we used the same restriction enzymes, it found the same restriction site every single time and cut it the same way. If you notice, when we mix these 2 pieces of DNA together, the sticky ends, these are called sticky ends because I have bases that are unpaired. So right here, I have bases that are not paired. And they will pair, right, when they're put together with their complementary bases. So when you mix these 2 pieces of DNA together, is because the sticky ends are complementary to one another, then they will form hydrogen bonds there, and they'll come together. So this is where our tape's going to come in handy here. Is we're going to tape together the sticky ends, and they should fit pretty well, and our bases should be complementary there. So when you have your recombinant DNA made, will you put your hand up for me, please? Henry, that better not be an insulin gene that you're tickling your chin with. Okay, we good? Awesome. All right, so now let's note some terms that you're going to have to recognize. Take a pencil, pen, I don't care, colored pencil, okay? On the inside of this, I need you to write R, like a lowercase r, DNA. So just in the middle, I think I just wrote, like, you know how we used to write like rRNA for ribosome RNA? I want you to write rDNA. rDNA. Because now this is what we call this DNA recombinant DNA, because it's a combination of 2 pieces of DNA from 2 different organisms. So that's what rDNA is. rDNA is DNA that has, again, just DNA from 2 different species. Recombinant DNA. So make sure you understand what rDNA is, okay? Now, we're not done engineering this plasmid. We have to put another gene in here that's an incredibly important gene. And it seems kind of counterintuitive, but we're going to give these bacteria that we're trying to engineer by putting this DNA in there, we also want to give them another trait. Yeah, they can make human insulin, but we're going to give them another trait where we have a certain antibiotic that will not kill them. So we're going to give them antibiotic resistance, and we're going to give them resistance to an antibiotic which is called ampicillin. Some of you have been prescribed ampicillin before because you had a bacterial infection. So I want you to grab the one that says AMP resistant gene. Okay, it's this bottom one that you colored in here. And just so that you understand, I want you to write ampicillin. Does ampicillin, does the name of it remind you of any other antibiotic that you've been prescribed before? Does it remind you of penicillin? Kind of sounds the same, right? Ampicillin, penicillin, okay? So I just wrote, just to make sure you understand, ampicillin, okay? And ampicillin is an antibiotic. And this gene is going to make it where ampicillin can't kill our bacteria that we genetically engineer. And we'll talk about why we want them to have that ability, okay? So once again, I said we're not done engineering this plasmid. We're going to insert another gene into this plasmid before we put this plasmid into a bacteria cell, okay? So let's kind of look down here. I'm going to use a different restriction enzyme. So a different restriction enzyme that cleaves at a different site, okay? So the restriction site for this new restriction enzyme is right here, okay? So it says it on your sheet here that the restriction site is CCGG. And when it finds a CCGG, it cuts between the two C's, okay? So let's open up our plasmid. We should find a CCGG, and you want to make sure, be really careful here, you don't want to cut in the middle. If there's a CCGG, I don't think there is. I think I took it out. We just have to make sure that we don't use a restriction enzyme that would cut like in the middle of this human insulin gene and put DNA in the middle of it, because then they wouldn't be able to code for insulin. The amino acids would be all different, okay? So it should be in the regular plasmid. Find a CCGG. There should one be high and low. When you do that on the original plasmid, like I have a CCGG right here, but it's going to be kind of different on yours. CCGG, you cut between the two C's, CCGG, cut here, okay? And all you got to do is open up that plasmid. And it's going to have sticky ends once again. Okay. And then for this gene for insulin, we need to cut it with the same restriction enzyme. You need to find two CCGGs. There'll be one high and low. You can see there's one right here, CCGG. CCGG, we have to go and cut out this piece of DNA so we just have that gene. Okay, and this is the case where it's got sticky ends here. Okay, and then just all I gotta do is mix the pieces together. And you'll see that the sticky ends are complementary. All right. So one thing that we have to watch out for, a couple things, again, when we're engineering DNA, is if we want them to make a protein, okay, and then we're adding several parts to this plasmid, we have to make sure that that gene stays intact that codes for a protein and certain traits. So we want to make sure there's no restriction enzyme or restriction sites in the middle of the genes that are coding for a protein, because then if we cut the middle of that gene, it's not going to code for a protein that we want our bacteria to make, okay? And the second thing we have to watch out for is the size of this plasmid, because if it's a really big piece of DNA, it can't come through those phospholipids into the bacteria cell. So it has to be a pretty small piece of DNA, a small plasmid, to be taken up by transformation by the bacteria cell. I was talking to, I kind of mentioned before, I was doing a parent-teacher conference with a guy who was a molecular geneticist. He was the one that did PCR before there was a machine to do PCR. And he told me, and I don't remember the exact thing that he said, but he was talking about how he was the first person ever to insert a plasmid, and he gave me the length of it. He said it was like 2,500 BPs long into a bacteria cell, which that was a big plasmid, and he was able to get it into a bacteria cell. And the 2,500 BP he was talking about was 2,500 base pairs. Okay. So again, we can't make this too big because it won't be able to go into the bacteria cells, right? So now let's kind of talk about what's going to happen. Oh, by the way, I do need to mention a couple tools here. So can anyone tell me in replication, DNA replication, remember the replication fork? We had an enzyme that could seal together the backbone between the Okazaki fragments. Can anyone tell me the name of that enzyme that could seal together the backbone between the Okazaki fragments? Mm-hmm. So we would need ligase to do this because our hydrogen bonds are going to form at the sticky ends, but there's still going to be like a gap between the backbone of these two nucleotides. So ligase can seal that gap. So ligase is a tool that we can use when we genetically engineer bacteria. Okay. All right. Speaking of a couple different tools. Oh, I didn't hear that. What was that? Yeah, it is. It's like the tape. Very good. Okay. All right. So if you look at my screen here, we have now, you can see the yellow recombinant DNA. So it's got a gene that came from a different organism. This is what our lab is going to do. I'm going to give you recombinant plasmids and E. coli, and you are going to get the DNA into the E. coli. I'm going to give you two tools. One, I'm going to give you some calcium chloride. The calcium chloride helps kind of interact with the phospholipids to create holes where the plasmid can make it through. So calcium chloride. And then the second thing we're going to do is called heat shock. So heat shock is basically if I was, and you can kind of see here, do you see how little holes kind of opened up in the plasma membrane and then the DNA can get through? Calcium chloride with heat shock, what heat shock is imagine if I put you in an ice bath for about 5 minutes. Okay, so you were in an icy water bath, and then I take you out of your icy water bath and I dunk you in a hot tub for 50 seconds. And then you get right back out of the hot tub and you get back into the ice water bath. I mean, you would probably go into shock. Okay, so that's why it's called heat shock. And when we do that, and since this is a lipid, remember, lipids can change consistencies when they're in cold and warm temperatures. The phospholipid bilayer gets changed a little bit and becomes more porous by heat shock. And so these pores develop, these little holes, and some of the DNA is going to make it in. Okay. All right. So now this cell can make insulin. But let's talk about the ampicillin resistant gene because that's an important part of this too. And let me go to my PowerPoint here. Okay. Why the ampicillin resistance? Okay. So the reason why we want some of them to be resistant to ampicillin, and I really wish I had this open. Let's see if I can open it and draw on it real quick. Why did you go down there? All right, now I can draw on it real quick. Maybe. Is because we're going to grow these bacteria. And we don't want to grow bacteria and feed bacteria that isn't transformed, that can't make human insulin for us. Where's my little draw thing? Okay, so when we take a look here, we just made, you can see it right here. This is the recombinant DNA. We have a gene that allows it to make insulin. And then we have a gene right here, which causes it to make a protein that destroys ampicillin so that ampicillin can't kill it. When we mix this plasmid in with bacteria, then here's what's going to happen is some of them are going to be transformed. Some of them will take in the plasmid, but then other ones won't. The DNA won't go into them. Now, when we take them, we suck them up with a pipette and we put them in a dish, a petri dish that has food. Notice I have ones that got transformed and other ones didn't. Now, one thing that we're going to put in this food dish for them is also an antibiotic, which is ampicillin. So the ampicillin is going to do this. It's going to kill all of the non-transformed bacteria, and then all we're going to do is grow bacteria that can produce insulin for us. So we're going to select for the ones that got transformed. Okay. All right. So a couple things before we end class for today. First of all, you do have a homework assignment to watch the rest of lecture 6.6. You guys were supposed to watch the first 30 minutes of it before today, and now you're going to watch the last half. You're going to learn more about restriction enzymes and some of the difficult problems that they're going to ask you about restriction enzymes. And for the rest of the class, we're going to kind of answer some of these questions together. So you should have another worksheet here that says 6.6. So this goes along with this gene splicing model that we just did. And let's knock out as many questions as we can in the next 5 minutes here. You guys will have to tell me what the answers are, okay? So it says, relate the terms below to the model that we just built and the actions that took place to build the model, okay? So what was the inserted human gene in our model? Insulin, the insulin gene. By the way, you can only answer twice, and then someone else in your class has to answer. So you can write in that in our model, the inserted human gene was the insulin gene. Okay, let's consider the plasmid.
The bacteria DNA?
Yeah, the original circular bacteria DNA. What was our restriction enzymes?
Ampicillin.
That is the antibiotic. This is our restriction enzyme because we found a site and we cut it. So our scissors represented the restriction enzyme that can cut DNA at a restriction site. Okay. What do we mean by sticky ends? And you might want to draw in. If I say sticky ends, what do sticky ends look like? So the sticky ends looked like this, right? They weren't cut straight through the double-stranded DNA. So this would be an example of what sticky ends look like. Okay, so what's the recombinant DNA? Does anyone want to say what we're writing down for the recombinant DNA? Yeah. Yep. This final larger piece of DNA that is a combination of DNA from two different species. Okay, what was ligase? Zach already hit the nail on the head with this one. What was ligase? Right. So it's kind of like the tape that fuses the backbone between two bases. Sorry, two nucleotides. Okay. If I say what was a restriction site?
Wouldn't it be the places that?
Yep. And so you could even write like GGATCC. That sequence is called a restriction site. And we had a CCGG was another restriction site. All right, so when I eventually see you guys in class, we are gonna continue on with this worksheet. We're gonna finish it and work on it, and then we're gonna and then we have another one, which is 6.7, which is all about restriction enzymes and the problems that they're gonna give you with restriction enzymes and cutting DNA and stuff like that, okay? All right, thanks for joining me for arts and crafts time today. You guys have a good day.